Welcome back. So far in the past few lectures, we have seen how drugs can be developed against enzymes, and so enzymes are the targets for new drug development. And here we looked at various aspects of inhibitors, the types of inhibitors and so on. And we also understood the mechanisms of how enzymes act and what kind of binding forces are present, and how we could mimic the active site in the enzyme, and for us to or we need to make molecules that can go and bind to the appropriate binding site in the correct manner, so that it can inhibit the enzyme. So, similarly, we have also looked at how receptors function and how certain receptors, most receptors are present on the cell surface, and they act as very important signaling hubs of signaling, and how we could develop new drugs against receptors. So, we also saw that we could have a very high level of signaling. So, in which case we will have to inhibit the particular receptor, or we could have very low level of signaling, in which case we will have to enhance the receptor's function, or we will have to increase the number of ligands. So, both of these approaches will require slightly different strategies, and so now we will look at the third major drug target, which is nucleic acids. So, we have already looked at how the major nucleic acids inside the cell are basically DNA and RNA. We have looked at the various types of RNA, for example, and how it can function inside the cell. And of course, DNA by and large is the primary storage of genetic information inside the cell, and so DNA becomes a very attractive target for drug development. So, although proteins are the targets for the majority of clinically useful drugs, like we looked at enzyme inhibition of enzymes, or enhancement of receptor signaling, or inhibition of it, all of these are proteins, and so proteins are the major targets. But nucleic acids are especially important when we are looking at antibacterial and anticancer therapy. So, in today's lecture, we will look at certain examples of how nucleic acids can be targets for drugs which are already in existence, and how we could understand how these drugs act in order for us to develop new drugs. The first major class of nucleic acid interacting agents are what are known as intercalating agents. Okay. Intercalation is basically a situation where a drug goes and slips between base pairs in a double helix. So, here is the Watson-Crick double secondary structure that is shown here, and there are, as we looked at, there are very important interactions that happen, and the nucleobases are interacting with each other through pi stacking, and so that creates a situation where, or a place where this drug can go and bind. Okay. So, these drugs, as you can imagine, would be typically planar, okay, with either aromatic or heteroaromatic features. Okay. So, once this gets in, then what happens is that it can then bind to the two different base pairs as shown here, and some of these intercalating agents also have charged species on the periphery. Okay. So, these charged species, which are positively charged ammonium salts, for example, can then have ionic interactions with the phosphate backbone. And lastly, we could also have van der Waals interactions as shown here with the bases. Okay. Now, some of these drugs prefer to approach the helix via the major groove, and others prefer to access via the minor groove. We have already looked at the major and minor groove in one of the previous lectures. Once they are inserted between the nucleic acid base pairs, the aromatic or heteroaromatic rings are held together by van der Waals interactions, and with the base pairs above and below. Several intercalating drugs also contain ionized groups, as we discussed earlier, and these can interact with the charged species. So, what in this process happens is that we have a very strong interaction, that is through van der Waals interaction as well as ionic interactions, and these can go inside the DNA, and therefore, they can form pretty stable species. So, the example that we are going to look at is Proflavine. Proflavine is one of the antibacterial compounds and under the major class of compounds known as aminoacridines, and these were used during World Wars I and II to treat deep surface wounds infections, and the drugs proved highly effective in preventing infection and reduced the number of fatalities. Right. So, Proflavine, as you can see, is completely ionized in pH 7. Okay. So, it has an amine, and so the amine can pick up a proton and form an ammonium salt, and therefore, it is completely ionized. And because it has a flat tricyclic ring as shown here, it can go inside the between the two base pairs, and once it inserts, again as we looked at earlier, it will form stable van der Waals interactions as well as ionic interactions. Okay. And after this insertion happens, the double helix becomes deformed. Okay. Now, after the double helix is deformed, what perhaps happens is, or what we could imagine that could happen, is that it would prevent the normal functions of replication and transcription. Another example of an intercalating agent is Dactinomycin. Okay. So, it was previously called as Actinomycin D. It is a naturally occurring antibiotic that was first isolated from Streptomyces parvulis in 1953. It has been shown to have an effect as an effective anti-cancer agent in childhood cancers. If you look at the structure of Dactinomycin, it has the planar phenoxazone system, okay, and it also has a peptide over here, which is a cyclic peptide. Okay. And the drug approaches DNA via the major groove of the double helix, and after it intercalates, the planar tricyclic system gets between the two base pairs, as we have seen previously. The charged amino group attached to the sugar is also important, and it forms an ionic bond with the negatively charged phosphate groups of the DNA backbone. It appears to favor interactions with the guanine cytosine base pairs, that is GC base pairs, and in particular between two adjacent guanine bases on alternate strands of a helix. So, you have G C interactions, and if you have two G's, then it prefers to bind to these types of DNA base pairs. The molecule is further held in position by hydrogen bond interactions between the nucleic acid bases and the cyclic pentapeptide, as we looked at earlier. Okay. The 2-amino group of guanine plays a particularly important role in this interaction. The resulting bound complex is very stable and it prevents the unwinding of the double helix. So, this in turn prevents DNA-dependent RNA polymerase from catalyzing the synthesis of mRNA, and thus it prevents transcription. So, the effect of Dactinomycin in an actively dividing cell is that once it goes inside and thus intercalates. After it intercalates, it forms an extremely stable bond with DNA. And after this complex is formed, because this complex is so stable, it prevents the unwinding of the double helix. And if unwinding of double helix does not happen, as we know from our previous lectures, that the single strand DNA is not produced, which is going to transfer the information to mRNA. And so, therefore, transcription is inhibited. And as a consequence, if transcription is inhibited, then the cell would stop replicating or stop dividing. The next example or we will look at is doxorubicin. It is among the most effective anti-cancer drugs ever discovered, and it belongs to a group of naturally occurring antibiotics known as anthracyclines. It was first isolated from Streptomyces puscitis in 1967 and contains a tetracyclic system where 3 of the rings are planar. Okay. So, again the common group that we see here is that it should have 3 rings and they are all planar. Okay. Doxorubicin also has an amino sugar. Okay. Here is a structure of doxorubicin approaching the DNA, and you can see here that this is the structure of doxorubicin. You can see the planar rings over here, and this is the sugar over here. Okay. and it intercalates using the planar tricyclic system. The charged amino group attached to the sugar is also important as it forms an ionic bond with the negatively charged phosphate groups. So, such as shown here. Okay. A number of experiments have been conducted to understand the intercalation by doxorubicin, and the data from these experiments support that once you remove the amino sugar, the activity of doxorubicin goes down, or the activity of the doxorubicin analog goes down. So, therefore, the amino sugar plays a key role in the activity of doxorubicin. Again, intercalation prevents normal action of an enzyme called as topoisomerase 2. This enzyme, again as we looked at earlier, is an enzyme that is crucial to replication and mitosis. So, the mechanism of topoisomerase 2 involves the formation of a DNA enzyme complex, where the enzyme is covalently linked to DNA. Again, recall, we have looked at this in one of the previous lectures about how topoisomerase 2 acts. So, when doxorubicin is intercalated into DNA, it stabilizes this DNA enzyme complex and stalls the process. Agents such as doxorubicin are referred to as topoisomerase 2 poisons rather than inhibitors, as they do not prevent the enzyme functioning directly, but what they do is that they prevent the by making a stable complex, they prevent transcription from occurring. The next class of DNA interacting agents are bleomycins. Bleomycins, as you can see, are a complex natural products, and they were again isolated from Streptomyces species in 1962, and they are one of the few they are among the few anti-cancer drugs that do not cause bone marrow depression. So, in many cases, because anti-cancer drugs target actively dividing cells, they cause non-specific action on the bone marrow, and so any drug that prevents or inhibits the growth of cancers, but does not affect the bone marrow is very useful. One of the key elements of the structure of bleomycin is this bis-thiazole ring as shown here, and this bis-thiazole ring is believed to be the major intercalating species. Once the structure becomes intercalated, the nitrogen atoms of the primary amines, the pyrimidine ring and the imidazole ring chelate an ion, that is ferrous ion, which then interacts with oxygen and is oxidized to ferric ion. Okay. Now, during this process, oxygen picks up one electron and forms superoxide. Okay. And superoxide can subsequently get converted to hydrogen peroxide, which then eventually forms hydroxyl radical. These together are known as reactive oxygen species, and the cell is very efficient at getting rid of some of these reactive oxygen species. However, since the generation of reactive oxygen species occurs near the DNA strands, this can result in the DNA strands being cut, particularly between purine and pyrimidine nucleotides. Okay. Bleomycin also appears to prevent the enzyme DNA ligase from repairing the damage caused, and whenever there is a damage that occurs on DNA, there are a number of enzymes that come and help in repairing the damage that is caused. Let us now look at some non-intercalating topoisomerase poisons. The anti-cancer agent Etoposide and Teniposide, they belong to a class of compounds known as Podophyllotoxins. These are semi-synthetic derivatives of Podophyllotoxin, an isomer of the naturally occurring agent called as Podophyllotoxin. So, here is the structure of the natural isomer, and here is the structure of Etoposide and Teniposide. Now, both these agents act as topoisomerase poisons. DNA strand breakage by this class of molecules is thought to occur by a free radical process that involves 4' phenolic group. And what happens, or what is proposed to happen, is that this 4' phenolic group forms a semiquinone radical, which then generates again reactive oxygen species. Evidence for this comes when If you remove this, or if you remove the if you are able to derivatize this oxygen into a methyl, right, then the compound becomes inactive. The presence of the glucoside sugar moiety also increases the ability of this molecule to induce breaks. The next class of next important molecule that we would look at is Camptothecin. Camptothecin is a natural product that was extracted from the Chinese bush Camptotheca acuminata in 1966. Camptothecin stabilizes the cleavable complex that is formed between DNA and the enzyme topoisomerase 1. So, by doing this, what happens is that it results in single strand breaks that accumulate in DNA. You may remember that topoisomerase 1 and topoisomerase 2 both nick DNA for their function. The difference between topoisomerase 1 and topoisomerase 2 is that topoisomerase 2 cleaves only one strand of DNA, whereas topoisomerase 2 cleaves both the strands. Okay. And so, since the complex between DNA and enzyme topoisomerase 1 is stabilized, the single strand break persists and it does not and therefore, this accumulates in the DNA, resulting in inhibition of proliferation. These single strands breaks can be repaired if the drug departs, but if replication is taking place when the drug enzyme DNA complex is present, an irreversible double strand break takes place, which then leads to cell death. Semi-synthetic analogues of Camptothecin have been developed as clinically useful anti-cancer agents. The next class of drugs that interact with DNA are quinolones and their fluorinated derivatives, fluoroquinolones. They both are synthetic agents and they both inhibit the replication and transcription, and they are antibacterial compounds, and so they inhibit replication and transcription of bacterial DNA. So, what happens is that they stabilize the complex formed between DNA and bacterial topoisomerase. So, once you stabilize this complex, as you can recall from the example of Camptothecin, it results in accumulation of breaks. Inhibition arises by the formation of a ternary complex involving the drug, the enzyme and the bound DNA. So, here is the structure of the quinolone, and this is the area that binds to DNA, and it also stacks, it is a stacking domain. This is the functional group that binds to the enzyme, and this also binds to the enzyme. The binding site for the fluoroquinolone appears once the enzyme has nicked the DNA strand and the strands are ready to be crossed over. At this point, 4 fluoroquinolone molecules are bound in a stacking arrangement such that aromatic rings are coplanar. The carbonyl and the carboxylate groups of fluoroquinolones interact with DNA by hydrogen bonding, while the fluoro substituent over here in position 6 and the substituent at C7 and the carboxylate are involved in binding interactions with the enzyme. Okay. Together, it prevents the enzyme DNA complex. Next class of molecules that interact with nucleic acids are alkylating and metallating agents. Alkylating agents, as the name suggests, are highly electrophilic compounds that react with nucleophiles to form strong covalent bonds. We have already studied in a lot of detail nucleophilic reactions in our introductory organic chemistry course, where you have majorly SN1 and SN2 type of reactions. Now, there are several nucleophilic groups present on nucleic acid bases. Okay. So, here are some examples. So, you have adenine, it has 2 of these which are nucleophilic. Guanine has this molecule which is quite nucleophilic, and cytosine again has this nitrogen which is nucleophilic. And now, N7 of guanine, for example, is highly nucleophilic and it acts as an excellent nucleophile with certain alkylating agents. So, drugs with 2 alkylating agents can react with a nucleic acid base on each chain of DNA, and what it might do is to actually to get a cross link. So, if it reacts here and then if it reacts here, here is your electrophile, and then it actually what it does is it makes a cross link. Alternatively, the drug could link to two nucleophilic groups on the same chain, that the portion of DNA then becomes masked from the enzymes required to catalyze the DNA replication. So, what could happen is you have a DNA strand here. So, it alkylates here and then it alkylates here, and then what happens is that that particular portion of the DNA becomes does not participate in replication or transcription. So, this might result in what is known as miscoding. Right. So, consequence of miscoding is that a slightly different protein is going to be synthesized, and this protein may not function the way the normal protein would function, and that could again result in stress, perhaps in cell death. The guanine base usually exists as the keto tautomer, allowing it to base pair with cytosine. This we have already looked at previously. So, when cytosine hydrogen bonds with guanine, you have 3 intermolecular hydrogen bonding that can occur. But once guanine is alkylated, as we saw in the 7th position, then it forms an abnormal base pairing, because you have the keto isomer here getting converted to the enol isomer, okay, or the enol tautomer. And so, the enol tautomer is actually more likely to bind not with cytosine, but with thymine. So, this again results in a situation which causes stress. So, such miscoding leads ultimately to alteration in the amino acid sequence of the proteins, because DNA is going to give rise to mRNA and mRNA is going to give rise to protein, right. So, if the sequence of DNA is normal, then a normal mRNA sequence would be produced, which is going to produce a normal protein. However, if you have some mismatch here, then it is going to result in a protein that is going to be perhaps different in its sequence. As we have looked at previously, the primary structure of proteins are very important in how they fold, and the folding is going to determine the function, and of course, it is going to also determine the structure. Unfortunately, alkylating agents, being such reactive molecules, can alkylate nucleophilic groups of proteins as well as DNA, which means that they have fairly poor selectivity and they have very strong toxic side effects. In some cases, they can even lead to cancer in their own right. Nevertheless, alkylating agents are still useful in the treatment of cancer. So, the first class of alkylating agents that we will look at are nitrogen mustards. So, these are related to the sulphur-containing mustard gases that were used as weapons in World War I. In 1942, the nitrogen mustard compound chlormethine, as shown here, was first used as medicinally, okay, and it is an alkylating agent, and as you can see, the nitrogen lone pair can do an intramolecular reaction, kick out chloride and form a aziridine type of ring. This, as we have looked at previously, is an example of ankymeric assistance or neighboring group participation. So, once this highly reactive aziridinium ion is formed, then it is a substrate for attack by guanine, for example, and once it reacts with guanine, it is going to form a covalent bond. Formation of this covalent bond is an irreversible process, and now the second chloride ion can be displaced from the resulting species, and once it is again going to form an aziridinium ion, another molecule of guanine or another nucleophilic base can attack and it can form a crosslink. So, there are some improved versions of these drugs that are used, such as cyclophosphamide, and we will look at this later in the course. The next class of anti-cancer agents which interact with DNA are lomustine and carmustine. These were discovered in the 1960s, and they are basically nitrosoureas which decompose spontaneously in the body to produce two active compounds. One is an alkylating agent and the other one is a carbamylating agent. Okay. So, here is the structure of these two compounds, and nitrosourea is basically this structure. Okay. Urea is this, and nitrosourea is here. Okay. So, let us look at the mechanism. So, what happens is that this nitrosourea undergoes decomposition to produce an isocyanate R N double bond C double bond O, and isocyanates are known to react with lysine residues of proteins. And once it reacts with lysine residues of protein, it is an irreversible process and it forms a urea derivative. The other part of the molecule produces this kind of a species, which can then generate nitrogen and hydroxide ion and produce a highly reactive alkylating agent. And this alkylating agent can then go and interact with DNA, produce a cross link. The next class or the next example of alkylating agent is busulfan. Busulfan was synthesized in 1950 as an effort to systematically search for novel alkylating agents. It is again an anti-cancer agent which causes interstrand cross linking between guanine units. Here is the structure of this molecule, and as you can see here, it has a very good leaving group in the form of sulfonates. And sulfonates, because of their excellent leaving group ability, can react with guanine, and once they are kicked out, they are going to alkylate guanine. Then subsequently another molecule or another nucleobase attacks and it produces a cross link as shown here. Okay. This mechanism involves the SN2 nucleophilic substitution and does not involve any intermediate such as the aziridinium ion. The next example of an anti-cancer drug is cisplatin. Okay. Cisplatin is one of the most frequently used anti-cancer drugs in medicine, and its discovery was actually accidental, and it was they were doing research on investigating the effects of an electric current on bacterial growth, and this happened in the 1960s, and accidentally it was found to have excellent anti-cancer activity. And again the mechanism here is that cisplatin structure is shown here, and it forms the equated cisplatin derivative, which can then interact with DNA to produce a very stable complex which inhibits the growth. Dacarbazine and Procarbazine are prodrugs. Okay. We will look at this example now, and these two compounds generate a methyl diazonium ion, right, and as an alkylating agent. Before we proceed, let us understand this term called prodrug. Right. So, when in organic chemistry we have studied the concept of chirality, and any molecule that has a potential group which can become chiral is called as prochiral. Right. So, similarly, extending the same analogy, we can understand prodrug as a molecule that becomes a drug. So, on its own it does not have any activity, but once it gets inside the cell or inside the body, there is some transformation that happens and makes the active drug. So, the anti-tumor properties of these compounds were discovered again in the 1960s after screening hundreds of compounds as potential antidepressants. So, here is an example as we looked at in the first couple of lectures, where we found that when we look for some drug and we actually come across a molecule which has an entirely different activity, this is one of the ways in which a new drug can be discovered. So, now let us look at the mechanism by which this molecule acts. The major metabolism inside the body happens in the liver, and a class of enzymes known as cytochromes P450 are very crucial for metabolism. So, this drug gets metabolized by cytochrome P450, and it produces this kind of an alcohol intermediate, which then rearranges and gives you a methyl diazonium ion. And this methyl diazonium ion produces nitrogen and a carbocation, which is methyl carbocation. And methyl carbocation, as we have seen, is going to be extremely reactive, and it reacts with DNA or with RNA and results in methylation. And once it gets methylated, then the DNA is destabilized, and then what happens is it eventually inhibits the proliferation. The next major class of drugs is mitomycin. Mitomycin was discovered in the 1950s, and again it has been isolated from the class of microorganisms known as Streptomyces. It is one of the most toxic anti-cancer drugs in clinical use, and it is another example of a prodrug. Of course, we will look at prodrugs much later in the course, and this is an excellent example of a prodrug. And what it does is that on its own it is really inactive, but what it is converted to an alkylating agent within the body. Now, the way in which this happens is that there are enzymes which can carry out what is known as a bioreductive process. There are many enzymes inside the body which can do oxidation as well as reduction, and these are known as oxidoreductases. Okay. So, when mitomycin encounters a bioreductive enzyme, this quinone here undergoes a reduction to form a diol. Okay. And now, quinone is actually an electron withdrawing group. We would imagine that carbonyl groups are electron withdrawing, whereas once it forms a diol, the diol is fairly electron rich, which can then promote this rearrangement as shown here to produce, eventually pick up a proton to produce a species, which then becomes the substrate for attack by DNA. So, as you can see here, the amine can attack over here, and this is like a Michael reaction, but it is not really a Michael reaction, but it is like a Michael reaction, and eventually forms this covalent adduct. One more round of loss of carbon dioxide and an amine generates the second site for attack by DNA, and this is how mitomycin can cross link. One of the important characteristics of solid tumors is that they have very little oxygen in the center of it. So, what happens is that imagine that this is a tumor and there is a large number of blood vessels that can supply the tumor. But since this is an uncontrolled growth, the generation of blood vessels is not uniform. So, this results in certain areas being here shown here, which have less oxygen because they have less blood supply. And so, therefore, when the drug gets into these kinds of hypoxic regions or oxygen starved regions, the bioreduction that happens is going to be somewhat irreversible. Otherwise, if oxygen is present, it is going to oxidize this back to the original drug. Now, what this helps us in understanding is that mitomycin is more effective in areas where there is hypoxia, and we will look at later, but hypoxic tumors are some of the most difficult to cure, because they have less blood supply, the drug also gets in much lesser, they respond less to radiation because there is less oxygen, and therefore, you have a very important problem that can be solved using mitomycin. Now, let us look at the next example, which is chain cutters. Chain cutters, as the name suggests, cut the strands of DNA and prevent the enzyme DNA ligase from repairing the damage. What they appear to act by creating radicals on the DNA structure, and these radicals react with oxygen to form superoxide and peroxy species, which then fragments the DNA.